meeting on Friday is all. Sorry, I missed the meeting on Friday. Is Sarah, Bitty, and Dave all speaking? I assume.
Quinn?
Yes.
Should I mute?
Welcome, everyone. We're getting ready to start this evening's program. It's 40 years of plant walks in Alton Baker Park, the plants, the people, and climate change. My name is Stephen Yeager, and I'm the immediate past president of the Emerald chapter. And I think I'll give a couple more minutes for people to get settled in, and we'll start with some announcements and introduce our speakers. Okay, I think we're getting settled in, and I think I'll get started here. I want to thank you all for coming to our program tonight, and I have a couple announcements. Let me get my screen fixed here. So I want to just announce a couple of Emerald Chapter events coming up over the next couple months. We have the Plant Lover Ramble, which is Sunday, December 13th from 2 to 4 p.m. This is a fun new event. We've had 2 months so far that we've conducted, and it's an informal plant walk. So rather than a traditional plant walk, it's a gathering of individuals, and we just share ideas, and we look at what's around us and look at what plants are there, and more casual than your more formal plant walk. And if you're interested in attending that plant walk, you will need to email Emma Lynn, and the email address is there on the screen. And it could be subject to cancellation due to the new governor restrictions, but we'll play it by ear. We know we have a new freeze on events for the next 2 weeks, but we'll see what the landscape looks like on December 13th, and we'll make sure to keep you all posted on that. We also, upcoming, we have our virtual holiday social, which we usually conduct this in person, but this year will be virtual due to the current circumstances. That will be Monday, December 14th at 6:30. We'll begin for a social half hour, and then we'll move into the traditional sharing of photos. So more information will be out on that soon, to where you can email your photos. And then this year, we're also going to add some contests, some plant trivia, some botanical inspired outfit prizes. And again, we'll be presenting more information on that soon. And then in January, we're looking forward to a talk about the Darlingtonian Wayside by Mary Santelman, which will be Monday, January 18th at 7 p.m. So we still have a full slate of programs, and we're looking forward to seeing you all there for those upcoming programs. I also did want to announce that this time of year, the nominations committee is seeking board representation for next year's election cycle. So if you're interested in getting more involved in a leadership role with the Emerald Chapter, we have positions open for the secretary position and perhaps other positions too. But if you just want to get more involved, yeah, reach out to Emma Lynn, and we can get you connected to or if anyone has any suggestions for officers or getting more involved with the board, we're open to those suggestions. And then also the Emerald Chapter is looking for a publicity coordinator. This is a volunteer position, but it will help the Emerald Chapter build a presence in the community. You know, they will work with the field trip coordinator and the program coordinator to publicize upcoming events. And we also are really excited about the new Emerald Chapter newsletter that we're putting together. And that'll also be part of the responsibilities for the publicity coordinator. Again, contact Emma Lynn, our current president, at mpresident@npsoorgan.org. So now I'm going to pass it over to Gail, who's going to introduce tonight's speakers. Take it away, Gail.
Okay, I'll just wait for the screens to readjust themselves.
Yeah, I think then Bitty, we should, transfer the screen.
You're muted, Bitty, so we can't hear you. My name's Gail, and I'm the program chair for the Emerald Chapter, and I want to thank you all for joining us while we make this little transition here. We're all still learning about the Zoom world and how it works and how we shift back and forth. But I just want to welcome all of you and also tell you that our speakers will address questions at the end. And those questions, there we go. And those questions can be put in the YouTube chat, and then they will be fielded. Oh, thank you, Bitty. They will be fielded by our president, Emma Lynn, who is working diligently behind the scenes, and your questions that will be put up, if you want, into the chat on YouTube will be fielded to the speakers, and they can address them at the end. So you can certainly type the questions anytime, anytime during the the talk, but they will be addressed at the end. So let's go back to that fantastic slide at the very beginning there. So I'm going to introduce the speakers, and there we go. And they're going to talk about this very beloved green space in Eugene, Alton Baker Park, which Bitty has shown this stunning picture of right here. So these are a group of 3 speakers for this one program, very talented group. And like many of us tonight, these presenters' love for plants began in their childhood. And not only their love for plants, but what time of year they could find their flowers. So that's a key theme tonight. And so for David Wagner, his love for plants started in the foothills of the Himalaya Mountains and continued into the Pacific Northwest during his graduate work at Washington State University. And then, of course, as he moved into Eugene as director and curator of the U of O Herbarium, that, of course, was during the time that he led the weekly Alton Baker walks, which he'll be talking about tonight. And now he's doing numerous things and activities. He's with the Eugene Natural History Society. He does a monthly column for the Eugene Weekly about time and natural history. And he has a consulting and research business, the Northwest Botanical Institute. And he was also recently recognized by the Native Plant Society of Oregon as a fellow. And that award is a very prestigious award in our society, and kudos and a well-deserved for Dave Wagner. So that's David. And then Bitty's love for plants started near Aspen, Colorado, and continued into Evergreen State College as an undergraduate, and on to Rancho Santa Ana in California, graduate work over to Zurich, Switzerland, and then back to Eugene in 2001 to U of O, where her research explored plants and fungi in local prairies and in the tropics. And although she retired this year, she's been continuing lots of research. She has a full plate. She's been traveling, all kinds of cool stuff. And so she's going to be talking about her role in the Alton Baker Park walks. And Sarah completed her wildlife degree at Michigan State University and has both undergraduate and postgraduate connections to Oregon here, to the Pacific Northwest, via the H.J. Andrews Experimental Forest, where she worked as a research technician. She completed her master's degree at U of O with Bitty, exploring plant phenology, and you'll learn that word tonight, and microclimates across the HJA watershed. Currently, she has a really cool title. It's called Super Bio, and at the McKenzie Ranger District, and worked on 2 recent fires, both the Holiday Farm fire and a fire in Colorado. So you see we have a pretty talented group here, and I'm going to turn it over to Bitty. Thanks a bunch, you guys.
It's Dave, actually.
Hello, this is Dave Wagner. It's a pleasure to be here and to talk about what work that began 45 some years ago and ended up growing. It's kind of like what botanists like. A small seed can grow into wonderful things. When I came to the University of Oregon in 1977 from Washington State, I did not know the flowers, and I thought one of the things a curator ought to do is learn the flowers of the area. And almost on a whim, I said, I'm going to go out across the river from the university, ride my bike across the Alton footbridge, now Frohnmeyer Bridge, lock my bike up and walk through the woods and see what's blooming every week. And if I did that every week, then I should be able to invite other people to do that. And that was an important part of this project, because thanks to the University of Oregon publicity, an interested group were almost always present at the beginning, and it kept up. And thanks to people who helped us out, like Tobias, Ed Alpson, Peter Zika, Bruce Newhouse, it ended up being done very, I will say, religiously. So that if I could not come, one of these people would substitute and keep track of what was blooming that week. And in 17 years that I did it, there was not one week and there wasn't something first out. There was a little bit of break when the herbarium was closed. And in 2010, Bitty picked up and carried on this idea of weekly plant walks, identifying everything that came out every week. And Bitty did more than me. She watched birds. That's where Peg came in. And so there were another 8 years. So that's how come from when it began to the data that we have now, it involved some 40 years of observation and long-term regular observations, which I didn't imagine at the beginning, turn out to be useful in many ways. And now one of the most important things, and this is where Sarah climbed in, is to see, can we correlate what information we have, because we have some hundreds of data points. What will tell us about what's changing, the climate and the area. Here's how the walk began. At the end of the Alton footbridge, about a mile and a half, walked along the what was the old Day Island Road, around what was the old radio station, and then back along the bike paths, always 1 hour. It was important first just to see what was there, and then when something was interesting, stand and look at it and see what we could learn. By the time we'd done this for a while, I made a plant list, and Bitty will show the sort of an example. This one was done by 1989. And so here you see some idea of what we handed out to people as they came on the walks. And it helped people to know what we were looking for. And I'll mention, for anybody who does plant walks, one of the most important things to remember is to number your list, because it's so much easier for people to find number 47 instead of looking through an alphabetized list and say, oh, that's Claytonia siberica. This is just, just a taste of the kinds of information that were put into the study that Sarah did. Now, I'm going to talk a little bit about finding, what it meant. What was the first bloom, and what was it that we recorded? That's just the first bloom of a species is a datum. And so here are the larkspurs, which are some of the most beautiful plants. The walk began in a relatively nice Willamette Valley bottom woodland, dominated along the edge of the river by willows and cottonwood, and further inland by oaks and big leaf maple. Now, so to look at what's the first bloom, the next picture shows what we were looking for. Up in the upper left corner is looking down the throat of one of these larkspurs, and you can see one stamen has stuck itself out and it's blooming. So the day in which we saw that marked the week of the walk in which the tall larkspur, the Columbia larkspur, started blooming. The next picture to the left showed that the development of all of those stamens shedding pollen lasted for a while. And in the lower right picture, having torn off one of the side petals, finally, sometime later down the inflorescence, the stigma started going out. So I have asked Bitty to put a picture up of a fireweed, which doesn't grow in the park. It would grow there. I've had it in my garden to illustrate the difference between the male phase and the female phase of flowering. Back when I started working on this, I wanted to coin some new terms because anthesis means that's when the flower's blooming. And so our definition of anthesis was anthers splitting and shedding pollen. But I like andresis for when it's in the male phase and gynesis in the female phase. A very famous botanist told me not to coin any terms. So it's male phase and female phase. So A is the first flower, the youngest flower on a stalk of flowers, a raceme. And you know, racemes are really common amongst herbaceous plants for the way they grow. They bloom from the bottom up, which means the top flower is the youngest. And up here at A is a flower that is just opened. If you go up the stalk, they're just buds. And the anthers are first starting to open up. And then you go down to B. B points to what's been happening with the female part of the flower. The style is coming out. If you look up at A, you'll see that the style is curved back behind the flower. In other words, that flower doesn't want pollen. And by the time you get to B, most of the anthers have been shedding pollen. The stamens are senescing and the style is uncurling. When you get to C, you're in the female phase of blooming. And so pollinators work from bottom up. It's just the way they work, so that every time they leave a plant, they go down and hide the predators. And that really, this interaction between how pollinators forage is a coevolutionary kind of phenomenon that determines what the flowers do, because flowers can't move, but they can control the movement of genes by who blooms when. And so this is called dichogamy. In other words, a bee comes in and it gets to the lowest flower that's presenting pollen at C, and it's brought in, that's got receptive stamens, and the pollen then fertilizes those stigmas. And as the bee goes up and collects nectar, it gets up to A, and that's the last one that it gets nectar from, and it gets a fresh load of pollen. And then it goes to the next flower, it starts at the bottom. And so this is called dichogamy. In other words, fertilization takes place in two ways. It's a very important part of breeding system. And so when it comes to saying, well, what's the first bloom? There's a lot more that we can learn. And probably the most interesting thing that we learned from these plant walks by going out every week was watching the big leaf maple. And they are marvelous plants. Now, these are big trees, and so they don't have just racemes, they've got hundreds of inflorescences on the trees. And what we found is that there are actually two different kinds of big leaf maples, two mating types. And here's a flower cluster of what I call type 1. In other words, I call type 1 were the ones that were female first. And you see it's got stigmas coming out. And you see that sterile stamen? I'll talk about that a little later, because the stamens in the female flowers never shed pollen. They really are female flowers. And in the type 1, you see all the female flowers were up there towards the base of the inflorescence, and down at the bottom, not yet out are male flowers. And in mid-season, these trees switch sex, and the female flowers start producing seeds, and then the male flowers come out. So if you go to the next slide, you'll see the type 2 trees. And this is the one that has all the male flowers first. So when we were recording first bloom, we recorded first bloom on these trees and not on the previous ones because they hadn't split pollen. And the distribution of flowers was different on these. This type, the male flowers are all up and down the flower stalk, not just at the tip. And there are unopened female flowers early in the season, right down towards the tip of the inflorescence. They're nascent, they're not out yet. And so when this one switches sexes at the end of its mating season, the male flowers are senescent and the female flowers are are receptive. And so I have a picture to show some of this because it's a little tricky. This is one that Bitty shows that the insects will cover the whole inflorescence if there are lots of flowers, but there are still some open flowers of the female. So here are type 2 trees at the peak of male flower with flies pollinating them. And there aren't many trees that we have that are fly pollinated. The next picture shows some of the detail for what we tried to figure out. So in the upper left is one of the type 2 trees. It shows that by the time the female flowers are coming out on the ones that had male flowers, you see they're all shriveled up. Female flowers are at the tip, and they will produce leaves, seeds at the end of the stalk. And the upper right shows a female flower, and I have put at the bottom showing the difference between the males and the females. The males are easy. They have no well-developed ovaries and styles and stigmas. They're just vestigial. The female flowers are deceptive, and they had confused a lot of local botanists. I did not know one botanist in the Pacific Northwest who had paid attention to this double mating pattern and switching sexes of the big leaf maple. Wonderful story. And so to sort of get it backwards, the next picture shows a sequence of a single inflorescence of the type 1 flowers. And on the left in April, by this time in April, the female flowers had already started to develop seeds, and the male flowers at the tip are shedding pollen. Come the 24th of April, the male flowers are senescing, the stamens are shriveling, and the female flowers are producing seeds by the wings. Come May, those samaras are developing beautifully. It's interesting, there are a couple of nice tri-winged seeds in there that are curious. But the male flower end, see, all the male flowers were at the tip of the inflorescence, and come June, the whole tip of the inflorescence is gone. And so if you look at the next picture, this is what led us to discover this, is that half the trees in the park had throwaway male flowers, and the other half didn't have. And what was the answer? It's heterodichogamy, 2 mating types that switch sexes in mid-season. Now we go on, as it turns out, pine maple will do the same thing. It's a little trickier. Because the next picture will wait a minute, sorry. There are some other phenological things which you notice, and I wanted to put this in to show what's happening, has been happening this last month. The big leaf maple, when its leaves start to senesce and pull all of their extra chlorophyll back into the plant, so they save their magnesium ions, because you can't have chlorophyll without a magnesium ion. There's a fungus that grows in many of the leaves, and what it does is that gives out a plant growth hormone analog and keeps the photosynthetic cells from around the fungus colony from decomposing, and uses those photosynthetic cells to give the energy to provide the tar spot fungus energy to make new spores. One of the things that we did as a group was to try to follow up on a study in England and Liverpool, where they thought this was pollution sensitive, because there were no tar spot in the middle of Liverpool and lots on the outside of the countryside. And we thought, let's try it in Eugene, different species. And what we found was that there was not much tar spot in the middle of Eugene, but the reason there wasn't is because we rake up the leaves. We break the life cycle. And that was something that as a group, we discovered before the people in Liverpool published on it. It was not pollution sensitive, it was sanitation sensitive. And so just quickly to look at the vine maple, most maples do have a breeding system, kind of like the big leaf maple. Vine maple does, but the way to understand it, again, is to pick the flowers and look at them closely. And the male flowers in vine maple, like the big leaf maple, have no developing ovary, but just stamens. And the female flowers have well-developed stamens which never shed pollen. So there's a young one showing the stigmas out, and then an older one showing the wings developing, just like the big leaf maple. Haven't studied the Douglas maple. There's a project for somebody. The other kinds of things that we look at, I just want to mention, is that so many of the flowers in our area do have separate male and female plants. The Oregon ash has male and female flower trees, and all of the willows do. The willows are interesting. When you look at them closely, you'll see again that there are male catkins with only anthers and female catkins with female ones, but there are nectaries in there. This is an oddball. It is enhanced pollination because insects come to get the nectar. Its petals aren't necessary to attract insects. The nectar is really an important one. The last one in this family that I like to talk about, that we learned about again from doing our plant walks. You start saying, okay, what's happening at this time of the year? And here is a stand of cottonwoods from along the river. This picture was taken on December 30th. No leaves on the trees at all. And you'll see there's a rookery, heron rookery in this tree on the edge. The next picture shows what it looks like. That picture was taken December 30th. This one here is taken on March 9th. And look at the trees that are right next to the ones of the rookery. They're almost completely covered with leaves. This is a remarkable feature that you don't get recorded by just noticing when the first tree blooms. You don't know how long the blooming takes. And with the cottonwoods, the blooming and leaf out lasts for almost 2 months. It takes a long time, so that the very first trees that are pollinating themselves, the males and the females, are completely isolated from the trees at the end of the season. What's its effect on the birds? I'm not sure. My last picture that I have to show you is of this same stand of trees taken on May 10th. And finally, the nests are hidden in the leaves. That's a question waiting to be answered. That starts from this kind of study of watching what's happening when. Did the herons choose a naked tree to build leaves in, build nests in, or was it accidental? I don't know. So there are lots of lessons that we've learned just by going out once a week and just watching what's happening now, what's blooming now. And I'm going to pass on now to Bitty, who will take the next stage because she picked up the next step in the search for what we can find by going out once a week and making little marks on a piece of paper as to what's out.
So this is Sarah. I'm going to take over.
Oh, okay.
Sorry, Dave.
Okay, it's fine.
So I did the data analysis portion of what's soon to be hopefully a paper, but the talk for now. So just to reiterate, so Bitty and Dave both did these observations. So we had kind of 2 time periods that we looked at. So from 77 to 93, and then from 2010 to 2018, we had observations of the week of first flower for over 200 species. So they just collected a lot of data. And so what I looked at was these 2 time periods, and then also how that all related with climate. So we took our climate data from a single point in Alton Baker Park using PRISM data, which is a model that was developed, I believe, at OSU, that where you can put in a latitude and longitude and get precipitation and temperature data for, I think, up to a century or more anywhere in the United States. So it's a very cool option to use. So that's where we got our climate data. And then we also downloaded data on the Pacific Decadal Oscillation, which I will explain in just a minute. We got that from the NOAA website. So I had 3 major questions doing this data analysis. So first, just because climate is such a obvious major driver in plants and how they grow and when they grow, I wanted to see if there was any differences in climate between 1977 and 93, so the period 1, and then period 2, which was the 2010 to 2018. And then we also wanted to know if there were any significant differences in flowering phenology, both within individual species and then across the whole group between those two periods. And then finally, what, if any, climate variables were predicting or were driving that flowering phenology over the entire survey record. So just before I get too deep, I want to explain what the Pacific Decadal Oscillation is. It's a phenomenon that's similar to the El Nino La Nina pattern. It's a little different in that it occurs, it's driven by sea surface temperatures, but they're temperatures that are in the northern part of the Pacific. And it's a much longer time frame, so typically 20 to 30 year phases with a little bit of variance in that. And basically what it is in what we call the warm or positive PDO phase, the sea surface temperatures directly off the coast of North America are warmer than average. And this correlates to warmer and drier climate, while in the opposite, so the negative or cool phases of the PDO, the sea surface temperatures directly off the North American coast of the Pacific are, they're cooler. So we get cooler and wetter weather. And you can see, so this bottom figure is showing the PDO phases over the last 100 years or so. And you can see in the blue, those are, it's a pretty long record from, I believe, the 50s until the late 70s was a negative phase, and then we had a positive phase, and then lately we've had sort of these shorter PDO ranges. So that was one of the climate metrics we looked at. So I just wanted to give a brief overview of that. This is just another kind of reiteration of the PDO. So the upper figure is Pacific Decadal Oscillation between 1976, which is right before the plant walk started, until 2018. And then these boxes on both figures are the different time periods that the plant walks occurred. And this actually looks pretty faded, but you can see it's not a direct pattern between precipitation and the PDO, but you can see that in years where there's a positive PDO, this bottom figure shows precipitation's difference from mean. So you can see that there was below average precipitation kind of between the 76 to 82 period, and that correlates to that positive PDO phase. And then the opposite occurs, 2010 to 2012 was a negative PDO phase, and we had above average precipitation. So then to get back to question 1, so seeing whether there were differences in climate between period 1 and period 2, I broke the climate metrics into kind of a bunch of different variables. So just in general, we had 3 seasons. So we considered the wet season started in November and went through March. And then we had the growing season, which was, or spring on these graphs, was April through June. And then the dry season was July through October. So plants don't use calendars. So we considered a year to be from November 1st until October 31st, because that's kind of a true phase for a plant. And so what we found was that between period 1 and period 2 for winter and spring precipitation, average winter temperatures, minimum winter temperatures, and maximum temperatures also for spring, we didn't see any significant differences between those 2 periods. There was some missing data for the dry season phase, so I wasn't able to do the analysis, but I would guess it'd be pretty similar. But I want to put in a caveat there that even though we didn't see significant differences in climate, just a reminder that climate records are typically compared over 30-year periods. So we have just over what's considered one entire climate record. So even though we didn't necessarily see significant differences in the data, that doesn't mean there's not a trend towards warmer and drier, which I think we're all pretty aware that that's what we've been seeing the last couple of years. So it just didn't show up in the data, but I believe that if we had a longer record, we'd begin to see that. So then for question 2, we wanted to see if there were any differences in phenology between period 1 and period 2 for the week of first flower. And so I did analysis of the over 200 species. We had data that fit all of the analyses rules for 112 species. And of those 112, we saw 25 species had significant statistical differences between the week of first flower between period 1 and period 2. But 14 of those were, it's 26 species. So 14 of those were advanced and 12 were delayed. So there wasn't actually like a marked pattern of everything, flowered earlier, everything flowered later. Nothing really fit any specific rule, which makes sense because there's 200 plus or 100 plus species that we looked at. So there's going to be variance in how they react. But we did see the 2 species that were the most different were Bellis perennis and Trifolium repens. So those are both weedy species. I don't have a figure here for them, but Bellis perennis had the largest advance where it flowered 4.6 weeks earlier in period 2. So over a month earlier. And then Trifolium repens was the largest delay where it flowered 2.9 weeks later. So again, almost a month, 3 weeks later. So those are really strong. That's a really big difference in that short of a time period. But so on this slide that you have, so I just picked 2 of the native species that we all know. So Berberis aquifolium and Oemleri cerasiformis. So Oregon grape and Indian plum. They both had significant differences in flowering phenology. And you may have noticed that the Oemleria actually has negative week of flower for the second period. And that's because when Bitty started doing the walk, she found that the first week in March wasn't early enough to capture some of these flowering times. And so they actually started going earlier, but they kept the first week in March as week 0. So that's why there's a negative number there. And then this figure on the right is just showing there's no significant difference in flowering times of the graminoids as a group or any herbaceous species as a group or the mean week of flower for woody species. Okay, so then finally, we wanted to know what, if any, climate variables were driving the flowering phenology. And sorry for this small text. So on the y-axis of all of these following figures, you're going to have the mean date of flower or mean week of flower. And then the x-axis or the bottom is going to have different climate variables. And I'm going to have to get you a little bit into statistics here. So because climate variables are all so correlated, so if you think, spring temperature and spring precipitation are going to follow the same pattern, we did what's called a principal components analysis. So in short, that basically takes all of these correlated variables and squishes them down and pulls out which variables are the strongest at predicting another variable, in this case, flowering time. And so that's called a PCA. And so on our PCA1, which is kind of the strongest piece of that squished down variable, it was primarily comprised of spring climate metrics, so precipitation and temperature, and then also Pacific decadal oscillation. And so what this graph is showing you basically is that when climate variables and PDO trended towards cooler, wetter weather, we had later flowering events. And when it trended towards warmer, drier weather, we had much earlier flowering events. And you can kind of really see that. So the symbol shape here is just the different periods. And then the color is actually the PDO. So you can see how in the upper left corner, which is the negative PDOs, that's that dark blue, is negative PDO indices. You've got later dates of mean flower versus when it's in the light blue and kind of trends towards that bottom right corner, that's going to be earlier dates of flower. And so this model was able to predict 43% of the variance in the data, which for ecology is a really strong predictive variable. We get excited about 5s and 10s. So 43 is awesome. And then this next figure, so because when we looked at that principal components analysis, so the squished down climate metric, PDO, the annual PDO index, was the strongest driver by far of that variable. And so we just wanted to see how just PDO was able to predict mean week of flower. And we got a really strong result. So this is, again, we've got PDO on the x-axis and mean week of flower on the y-axis. And that dashed line is a line of best fit. So you can see the data is a little more spread out. It's not as tight of a predictive model, but it's still able to predict 23% of the variance in the data, which again is a really strong number. And just to note, this slide and the next slide, the color is year, not PDO anymore. So you can see there's really no pattern in year how it relates to flower. The colors are just kind of scattered all over the place. And then finally, this species, so we had Prunus virginiana, had the strongest relationship between PDO and mean week of flower. So I looked at all 117 species with both, or 112 species with both the PCA1, the principal components axis, and PDO. So if you want to read the paper, that'll be in there. But just to give you a quick blurb and see how strong this is for some of these species, PDO was able to explain 49% of the variance in when bitter cherry flowered. And you can see there's a really tight fit of the data to this best fit line. So that's about what I have. Bitty, I think you're muted.
When I'm in screen share, I can't unmute. Okay, did you guys unmute me somehow? Thank you. When I was in screen share, I can't unmute. And I can't make it back up anymore. There we go. Okay. So can people hear me? I hope so.
Bitty, we can hear you now. You sound great.
Yeah, sorry. When I'm in screen share, I can't unmute. Okay, so what Sarah was talking about with the small sets of data, we didn't see any big differences in drought. But if you look at the full data set, so this is drought data from NOAA from 1895 to October 2020. And the upper part that's green, that's when it's not in drought, and the orange is when we're in drought. And what you can see is that over the last, basically the last 20 years, we've mostly been in drought, and the precipitation has been getting lower over time. But when we just compared Dave and me, we couldn't see that because there was too much variation in the data. But this is important because drought is causing the trees to die in Alton Baker Park. Not all the trees are dying, but the conifers are. And the conifers probably are a little bit out of range in the valley bottom. It's a little hot and dry for them. So we're losing a lot of the incense cedars in the park. And they're also showing up with many pathogens. So on the right is a picture of Gymnosporangium rust fungus that shows up on all the trees the year before they die. I think the fungus knows what's coming. And drought means that fires are going to be more likely in Alton Baker Park. This is a picture from KMTR from last July, and a small area burned, 2 acres. But we really recommend, based on other work, that the city prepare a fund for native seeds, and that when there are fires, they broadcast them on those fires. Because the grasses in the foreground are not native species. They're almost all invasive. It would be really good to start replacing the invasives with natives again. Here we're looking at 2 images on the left are from when Dave started the walk in 1977, and an aerial view in 2018 of the same place. So this is the area where we do the walks. And you'll notice in the picture from 1977, there's what looks like a rocket landing pad. Well, that was a gravel pit, and it was filled in about the time that Dave started the walks. And between the period of 1978 and 1993, they picked up Calandrinia ciliata every time they did, every season. But when I started in 2010, it was not on the walk anymore. And this is the simple result of succession. Its habitat no longer existed. There wasn't the open, sandy, gravelly soils anymore. It was covered up with other plants. Another case of change, of succession that you can see is that the hazelnut orchards are smaller in extent than they used to be. And if you look in the lower picture, the 2018 picture, you can see that you can't really tell that there's rows of trees anymore because their canopies are now interlocking. And if you look on the right, this is a photograph from underneath that grove of hazelnuts. And you can see that it's now quite a nice place for native plants. And I will add that these native plants would not have been there except for the fact that Nearby Nature started a little nursery of perennial plants in there, of native plants, and they've taken off and done really well. And I actually recommend that this is done more often, because once again, the natives can't get there anymore because there's so few of them. Another successional change was that it just became shadier overall. In the square in 1977 versus 2018, you can see that the canopy has filled in tremendously. And this meant that we lost some species that like a little bit more sun. For example, Dave and I measured the same exact Philadelphus individual during all of our walks. We had exactly one on the walk, but it died in 2016. And it died because it was too shady. And finally, the last successional change I want to talk about is one that is fairly concerning. In the 1960s, 3 major dams went in on the Willamette, on the upper Willamette, and the flooding of the river really diminished after that. And cottonwoods are dependent on flooding for regeneration because their seeds germinate best when they land on recently scoured gravel bars, basically, where it's still damp. And some students and I did a little survey along the banks of the Willamette a couple of years ago, and we found that there were no young cottonwoods. Now, our cottonwoods can live a couple hundred years, so maybe those dams will break and we'll have succession after that. But again, it's something the city might need to think about, is that the cottonwoods are going to start dying off. Okay, another big change that happened over the 42 years of this study is a lot of invasions. Essentially, we saw 2 or 3 new species each season that we did the walks. And of our 271 species, more than half of them were non-native. And if you look at this picture, only the trees in the background are native, literally nothing else is. And then since Dave stopped the walks in 1993, we've had 24 new invasions. And I'm going to talk about the 5 most dangerous of those now. Okay, so shining geranium, I think I first saw it in 2010 and I let the city know about it. It was a very small patch near one of the benches in the field, and they promptly got on it. Here is Ted, he's flaming it. But the problem was that they mowed, and they still mow every year, the edges of the roads. And you'll notice in the picture on the right that there's scraped ground. That's because the mower was set too low. And every time you scrape ground and cause disturbance, it spreads weeds. And then if you have a mower that's picking up those seeds, you just spread them everywhere. So right at this point, shining geraniums throughout the park, and it's largely the result of mowing. Another invasive plant that I've spent far too much time working on is Brachypodium sylvaticum. The photograph on the left, the big one, was taken not in Alton Baker Park, and that's good because the city's doing a good job of trying to control Brachypodium. They work on it every year, and it doesn't look like this, but it could. Essentially, our forest did not co-evolve with lawns, and this is a shade-tolerant grass, and it grows extremely well under forest canopies. And it is also spread to a large degree by disturbance. And on the photograph on the right, you'll notice a rogue trail. That's where people get off with their mountain bikes and do a little jump. Well, you do things like that, you make rogue trails, you spread weeds, and that's certainly happening in the park. Okay, I want to talk about 2 species that I think are particularly concerning. These are evergreen shrubs to small trees. On the left, you have Prunus laurocerasus, the cherry laurel, and on the right, you have Prunus lusitanica, the Portuguese laurel. They're pretty easy to tell apart. The Portuguese laurel has much thinner leaves, and the cherry laurel, the one on the left, has fairly substantial leaves with this quite strong yellow venation pattern. So I first saw them flowering in Alton Baker Park, the cherry laurel in 2010 when I started, and the Portuguese laurel in 2015. Notice that the first herbarium specimens of these were 1933 for the cherry laurel, and that was collected by Henderson, who some of you will know, on the U of O campus. And I am concerned that it's quite possible that the U of O campus seeded our initial plants of Prunus laurocerasus. They're bird carried, and so if people plant them in their yards, they're going to make it into the wild. And the Portuguese laurel, on the other hand, didn't show up in an herbarium specimen until 2003, so it's a more recent invader. The last species I'm going to talk about is the one I'm, quite frankly, the most worried about at this point in time. This is Ranunculus ficaria, or Ficaria verna, depending on your taxonomy, Lesser celandine. And I first found it on our walk in 2018. The upper picture on the left, you can see the plant kind of in the middle on the left. And there's a closer up picture of it. And I let the county know about it. Let the city know about it, and it's still there. This is why we need to be worried. This is not from OSU up the road. This is from Ohio State University. This population resulted from 2 gardens, and this is what happened in a 40-year period. So this plant completely covers the bottomlands along the river in this part of Ohio. So it crowds out everything else. Nothing else can grow because this is the only thing that does. And this is what we will lose. we'll lose the yellow violets, we'll lose the trilliums, we'll lose the candy flowers and the fringe cups. And I think that's a really big loss. There's lots that we can do. I really, really recommend that everybody start using iNaturalist. And when you see things like this Ficaria, if you take a picture with your phone and upload it to iNaturalist, it comes with coordinates, and then we can go out and find it and get it. So this is a tool that the city can use to find where these plants are. So I strongly recommend people use that. I also think that the Native Plant Society needs to do some partnering with the city of Eugene, in particular to do some work at Alton Baker Park. On the left, there's a photograph of one of my master's students, past master's students, Jesse McAlpine. He and Tobias and Friends of Trees and a number of other organizations did a couple of days where we used weed wrenches, which is what Jesse's carrying, to wrench these woody invasives out of the ground. And in the wintertime, you can see they're really easy to see because they have evergreen leaves, which is part of why they're dangerous. They shade out the understory. Everybody needs to ask nurseries to stop carrying invasive non-natives. And I would say that these 5 that I've just highlighted would be some of them. 3 of them are commonly carried in nurseries. The 2 prunus species and the Ficaria is also sometimes at nurseries. And we also need to work on a state level ban, just like we did with ivy. We need to do that with, I think in particular, these 2 woody prunuses and a couple of others. And if you really need a hedge, wild roses make good hedges. Okay, so I'm done. And I think we're ready for questions. So I hope people will ask them.
Hello, this is Emmeline. I'm the current president of the Emerald chapter. And thank you so much for that super interesting presentation. Let's see what we got here in the chat questions. Let me pull that up right now. And I'm not seeing so much yet. Anyone who's listening to this right now can go ahead on YouTube. Feel free to enter your questions in the chat.
I think for me, the oddest thing about giving a talk is that the questions are the part that I like the best. And it's harder to have that interaction with an audience when you're talking to a screen.
Yeah, definitely. It's a little tricky. We're adapting the best that we can. Let's see. Okay. Well, I got our first question here from Amy Desert. And it is there anything a person can do to eliminate shiny geranium on your property? I have pulled a bunch up, but I have 2 acres of it.
I have personal experience with this. We just bought a house in the South Hills we've got a 1/3 of an acre with shiny geranium everywhere. And I bought a propane torch at Home Depot and relentlessly torch my geranium every November. So I've also tried the black plastic method, and that seems to work pretty good too. Just covering up when it's hot and sunshading it.
Yeah, so one trick with shining geranium is that every time you pull one, you disturb the soil and you bring up more seeds. So the trick is to avoid disturbing if you possibly can.
Okay, great. So we have Larry asking, is NPSO active in following up on these recommendations? And I guess if it's okay, I'll take this one. As Stephen mentioned at the beginning, we are actively looking for more folks to be involved in our board. And another option to being involved in NPSO is being involved on a committee. And one of our committees is the Conservation Committee. It has not been very active, but I think that it would be great and exciting if there were more folks in the Eugene area who wanted to get active on the Conservation Committee. And I certainly think that some of this work, working with the city and working with the state on some of these native plant and invasive species issues is definitely exciting stuff. So Larry, if you're interested in that, go ahead and email m_president@npsorgan.org and we'll get you in touch with the Conservation Committee. If anyone else has, who's on the Zoom, if David or Bitty or Sarah or Gail or Stephen, you have anything you want to add to that, go ahead. All right, and so another question, are there any projects in Alton Baker that are currently underway to control weeds and plant natives?
That would be a really good question for the city. I would talk probably with Emily Steele, maybe. And also Nearby Nature is quite active. They often have work parties. The Willamoot area, though, the part where Dave and I do our walk, really could use some attention, and it's a little far from Nearby Nature, so they don't get out there.
All right.
There is a citizens committee for Alton Baker Park. But as far as I know, there are no biologists on that group now, so they may not be aware. A conservation committee would be a good thing.
Okay, great. Well, I put the email for reaching me, the Emerald Chapter President, in that chat. And if folks are interested, Native Plant Society of Oregon and Native Plant Society of Oregon Emerald Chapters and all volunteer organizations. So it's definitely on us to get involved and do this work. And moving on to some more questions, I see Pamela Swisher wants to know, would any of the speakers like to comment on the spread of bedstraw gallium everywhere, but I've particularly noted it in Hendricks Park?
So are you talking about the native gallium or the non-native ones? We have a native gallium. It's weedy, so people don't like it, because it tends to grow where they don't want it, but it's native. Are you maybe talking about the sweet bedstraw? The wider leaved one?
But Pamela clarifies and says the non-native gallium.
On Skinner Butte?
Yeah, Hendricks.
Oh, yeah. Don't know it. Haven't been up there for a couple of years. Asperula, isn't that woodruff? Sweet woodruff is in the family, and it does spread. It's a common. There are a lot of garden escapees, which tend to slip under our notice. Like the cuckoo pint, the arum, which is spreading in our neighborhood, down by the Delta ponds. Hasn't gotten onto our watch list because it's spreading slowly.
Yeah, it's just another warning to if you plant something that's not native and it grows really well, it could get out here, because plants seem to do well here. It's more reason to plant native whenever you can. But I don't have any specific advice for the bedstraw.
Yeah.
So we have a couple more great questions here. We have JTube4 has some great questions, starting with, how much do you think the phenology results at Alton Baker reflect what's happening all along the Willamette?
You want to take that, Sarah?
Yeah, sure. Well, I'm not sure I can speak exactly to the Willamette, but I did my master's research at the H.J. Andrews, so up in the Cascades. And we saw a stronger pattern of advanced phenology there. So I think I would say that because the Alton Baker, so many of those species are weedy. They're incredibly tolerant, incredibly resilient plants that are going to be adapting to their climate. And they're just more flexible, so they might not necessarily react as quickly with flowering time. They're just more resilient. So our natives, I think, are more strongly affected by these shifts in drought and these warmer, drier springs that we're seeing. So I did see that in my master's research at the Andrews, that the species at higher elevations that are already on kind of the range of their growth habits are going to be more strongly affected. So they're going to, we saw shrubs advancing like 2 months, flowering and leafing out 2 months in advance up at high elevations when we had that 2015 super drought year where we never had snowpack. I guess that sort of answers it. But I think that Alton Baker, you kind of have to split it out into the natives versus the non-natives more specifically to see how the overall Willamette is trending. But I do think Alton Baker would be representative on the native scale for sure.
So the next JTube4 question is, what's the biggest threat to native plant diversity within Eugene? Climate change, invasive species, land use change, or other?
Yes.
Wow.
And that's a tough one.
You know, it's kind of all of the above. Invasions are pretty bad. You know, at least in Alton Baker Park, I think we've seen more loss as a result of invasives than we have of climate change so far.
But also climate change, I think, exacerbates our invasions.
Exactly.
And land disturbance exacerbates our invasions. So.
And you know, I mean, yeah.
The other thing is just, you know, I mean, like development is a real issue. Having been here for almost 50 years, and just across the river from north of Valley River Center, when I first came, those were pastures and woodlands. Mm. I would ride my bike over there and you know, and gather material for teaching. Now the pastures were already changed, and this is one of the things is that so much of Willamette Valley was turned to agriculture, and that was like before our time. And now, you know, what breaks your heart is when the agricultural land is turned into condos. And so the diversity that had been there before, what had been there, it all that's always been a global problem. So one of the things to do, I guess, is to pay attention to those little fragments of nearly natural areas, wherever they are in the parks, recognize them, and you know, and manage for them. When I first got on a Parks Advisory Committee, the idea was that if you didn't do anything to an area, it was a natural area. And that's not true. If we want a natural area, we have to manage for it. We have to pay attention to it. And just paying attention makes a big difference. I'll point to the Friends of Buford Park, which is a group you know, dedicated to just maintaining what little bits of natural area that is up there on Mount Pisgah. And so there are these little fragments, just paying attention to those. And then the invasives, it was because there's some things that, well, like ivy is going to be everywhere. And if and that was one of the things about the Wheelamud area that got it started. When the ivy first crept in there, it became clear right away that that was something we could do something about. And for a good many years, the area right around the north end of the Otson Footbridge and that sort of woodland that was in there, a group of volunteers managed to keep the ivy down. But it has to be done, and it looked better. But going through one ivy pull doesn't take care of it. Yeah, there has to be an annual regular return. Doesn't take as much as it does the first time, but it's the ivy is a real enemy. I watched it smother native plants, so that you can do something about it. And I'll give a shout. I don't know where Jerry Morcello is, but there was a good part of that area, that one guy, bus driver for the city of Eugene, who kept it clear of ivy for years just by himself. Permission to work with the city, but that's a possibility. Some of these, adopt an area and stick with it.
I would also say adopt your garden and plant a lot of natives in your garden, because some of our best hope now is what we do, and if we plant our own gardens with natives, we've made a lot of birds and insects happy.
Yeah.
Any other questions?
Yeah, so moving on to a little bit of a different topic, Landacre wants, or let's see here, where was it? I'm sorry, J24 wants to know, have you observed, even just anecdotally, changes in pollinator activity over the same time period, or bird breeding changes?
Well, I can say that we only started watching the birds in 2010 or 11 maybe. And it's sort of too short of a record to be able to say. But I will say that Alton Baker Park gets a lot of birds. And during the spring, they don't care if it's 12 o'clock or 1 o'clock. And without the leaves on the trees, you can see them. In other words, you can see birds later in the day than you normally do in the summer. And so if you go for a walk at noon during the springtime, you can see 30 species, not a problem. In terms of insects, I have to say I was a little bit too focused on finding all 271 of those plant species. So I wasn't paying as much attention to the insects, but that would be a really great project for, say, a new faculty member to start doing. And we do have new faculty members who might do that.
And I would, part of this too is that there are not an awful lot of projects that do carry on. These were, what we've done was essentially voluntary. Like there was no grants.
Exactly.
Go out and watch those. And the problem with grants and support is that they've got a grant period and then you have a deliverable. And so I'll go back to that the people who came and were the citizen scientists, the ones who helped, people can do that. It just it's like deciding that you're going to adopt and watch, and maybe some kind of interaction sharing through the Native Plant Society, so that once a year or so you get together on Zoom and say, here's what I've seen in the little patch of native vegetation that's down by the river behind the Fred Meyer or something like that. But I live next to it.
Yeah, keeping records is really it's remarkable how you can extract a lot of information if you've written it down, but if you don't write it down, you won't remember. And so taking photographs and writing things down, then 20 years later, somebody might be able to see a trend in that data.
Yeah, I'll put a plug in here. I believe it's called Nature's Notebook. Is a citizen science phenology observation website that has data sheets, and you can enter in plant observations or bird observations, and then researchers can use that data. And it's nationwide, or it might be worldwide, but I know it's all over the United States. Oregon State has tried pretty hard to promote it for K through 12 students, but it's a really friendly, pretty great way to do phenology records. If you get a wild hare to start doing stuff like this, it's pretty nice. So, and it's fun to watch. You pay a lot more attention to how things change when you deliberately think about it. So.
Awesome. So Beth Stein from Nearby Nature hopped on the chat and wants everyone to know that they have volunteer and intern opportunities for anyone interested in native plant restoration. You can check them out at nearbynature.org or email info@nearbynature.org. Again, that was Nearby Nature is a great way to get native plant restoration in the park going on. And it sounds like they regularly have opportunities. So check them out. That's a great resource. Well, are you three up for a few more questions?
Oh, yeah.
Okay, let's see. We have Landacre would like to know your thoughts on ex situ conservation of plants that sites are being invaded or degraded. And I'm not familiar with that term. I'm wondering if that's in situ.
I'm wondering if she means removing the plants in order to conserve them and putting them somewhere else.
Yeah, but two things there. One is to go into an area where there are very few things, gather some seeds, and then try to propagate them off-site. And that's what ex situ means, as opposed to going into an area where there are very few plants and doing what you can to remove obstacles to their survival in that area. And that's in situ conservation. And they're both strategies depending on species and available resources. In general, the people in the endangered plant community have avoided encouraging ex situ by saying, okay, you don't have to worry about this population because the Berry Botanical Garden is propagating them and they've got hundreds of plants and the species is not in danger.
But the minute you do that, you have removed it from its natural context, and the pollinators and the birds and the fungi, everybody always forgets about the fungi, but they're all associated with those plants. So you take them out of their habitat and you've broken the web.
That's why in the Endangered Species Act, one of the most critical parts of it was the critical habitat designation and protection. Not just the species, but the critical habitat. Because any conservation ecologist says you don't protect species, you protect habitats. And if you protected the habitat, then the species that grow there are being paid attention to. I wrote an essay 20 some years ago where first time I saw these meta-analyses of population growth and peak population. And the idea of peak population had never crossed my mind. I just sort of thought that the Earth's population was going to keep growing until the Earth exploded. Well, it won't. There will be people that make it through. And that's when I thought, this is where they're not all going to be like me or us. So I'm going to put a lot more energy, and this may not this may be a little counter to what we're saying, but I put more energy into preserving as much as possible of natural habitats rather than just trying to get rid of invasive species because they're invasive species. It would save the best we have for the next generations.
Yep.
And oh, go ahead, David.
Oh, I just, that's why, I'll give a plug for Nature Conservancy, one of those organizations I've been involved with for decades. It's their job.
Yeah.
And the last question I see here on the chat is regarding invasives, has any distinction been made to type of habitat, open prairie versus riparian forest, for example? And that's coming from our Paul Gordon. And I'm interpreting that as in terms of your experience with invasives in the park invading those particular habitats.
So I guess that I've been, I think the most native rich part of the Willamette part of Alton Baker Park is the bottomland forest. And I am concerned about it because of these non-native evergreen prunus species that will shade out the understory. We just don't have, at this elevation, the rhododendron's not very common and it's not very shady anyway. So we just don't have anything that lives in the forest that casts that much shade. And in Alton Baker Park, there's almost nothing left in the, that's not quite true. There's very few native grasses left. You have to know where to look. There are, I found my patches. And that's part of why I'd really like to see more native species spread after fires, because there's no seed bank anymore. And so if we're going to encourage natives in Alton Baker Park, we need to take advantage of those fires and let some natives take over. So I don't know if I answered your question, Paul, but feel free to email me.
In general, open areas, grasslands are much more difficult to manage as native species, and the experience goes back to the people in the Midwest, the prairies there. In order to bring back those prairies, they have to do it practically square inch by square inch. Whereas if you've got a woodland, it protects its own species better. It's easier to protect and conserve and restore a woodland than it is a grassland. And I'm pretty sure that's generally true, at least across this country.
It's generally true because there's fewer species that are shade tolerant. And notice that all 5 of the species that I talked about are shade tolerators. And that's part of why they worry me. Because our forests are in better condition in general.
Mm-hmm. Because when and that's the thing, is that invasive grasses really have an advantage, an ability to take over a grassland if they're adapted to that habitat type. And that's especially in Eastern Oregon, this reason why cheatgrass has taken over hundreds of thousands of acres, and it's really hard to recover from that because the native grasses can't restart if all the water has been pulled out of the ground by the grasses that cheat them.
Yep. Well, thank you, everybody that I can't see.
Yeah, thank you. Are you up for one more question?
Sure.
This can be your last question if you want. We have a question, which is something you didn't touch on. Margaret is wondering about the poaching of native plants in Alton Baker Park?
I can speak to that a little bit. So we've had trouble mostly with our berberis, with the, give me a common name, somebody.
Fall Oregon grape.
Oregon grape.
Fall Oregon grape, yeah. We lost a bunch of it a few years ago. And the problem is that these are, we have essentially old growth stands of berberis, and the bush tits nest in them, and they're really important for early pollinators because they flower early. And they're being wiped out by people who are collecting them for their roots for herbal remedies. And darn it, grow your own. Stop it. Wild harvesting, when we have so few of these native populations left, is really dramatically awful.
There's a variety of people who do this. I have a friend, Howie Brownstein, who is an herbalist. And one of the reasons we got along is because he was also a conservationist, and he has taught his students, yeah, you have to collect these things, but you don't have to destroy them. Yeah, one of the reasons that I did so much exploring for teaching taxonomy is because I wouldn't send the students out to collect until they knew what they were doing. And I could easily, every week, give plenty of material teaching in a class without seriously damaging a population, because I knew where I could get just enough for that lab and know that I could go back and it would still be there year after year. In other words, I sort of thought that if I were an elk, I won't do any more damage to this than I would if I was an elk.
I will also point out that it is illegal to pick plants in any of the city parks. So if anybody needs that incentive, if they need to know that, it's out there. You can get permission. Like I got permission when I was teaching systematic botany, and there were a few things that I would get in Alton Baker Park, but I had permission to do it. And again, I was very careful about what I did. So if you see people picking plants in Alton Baker Park, just like if they're walking their dog without a leash, you can say something.
Yeah, that's true. There are limits. It is permissible to get native plants in the national forests, but you're supposed to get a permit. And in the process of getting that permit, you're supposed to learn how to do that without ruining the landscape. I mean, if you want some, there's ways to do it. If something grows in a ditch that's plowed every year, that's a good source, rather than going up and getting at the bottom of a talus slope that's stable.
Yep. Great questions, everybody.
Yeah, thank you. So Bitty, I have a quick question out of curiosity. Are you allowed, so obviously it's illegal to pick native plants, but what about non-native plants? Like, for example, if I come across a patch of celandine, can I pull it up and take it away from the park?
So the answer is that you really need to notify the city.
Okay, thank you for clarifying that. That's good to know.
Yeah.
If somebody else sees you, they don't know that you.
Right, absolutely.
Exactly. I mean, and also, I mean, we have really amazing people who work for our organizations, and they're generally pretty responsive. But I do think that the Native Plant Society could help a lot, because there's so little money now that, they've given up on celandine, for example, and yet you saw what can happen. Mm-hmm. And, if we work with the city, we can help. Yeah.
All right. Well, there are, it looks like, a few more questions, but it is up to you. And if you're ready.
Ask them away. Ask them away.
Okay. So we have a question from Alan Pittman, wonders what you think of the city planting all the giant sequoia.
I signed a letter against it.
Not my favorite.
Yeah, I'm not in favor. Were you in favor, Sarah?
I get the idea, for carbon sequestration, but we have plenty of native species. I mean, the sequoia is not native to Oregon, and I think oaks and prairie species would have been more appropriate. But I applaud the general idea that I definitely think that we could have had some more natives included in there.
That although there is another question that comes up with there, how native does it have to be?
Yeah.
Yeah, like I planted some twinberry in my yard, and a friend of mine who's a purist says that twinberry isn't supposed to grow in the Willamette Valley. Well, it probably used to, still does at the coast.
And I think the idea was that we'd have a climate resilient species. The sequoias are from Southern California, but there's some places where there's sequoias that are 7 feet apart, and like they're right next to each other.
They said they weren't going to do that. I mean, this was an issue. I mean, when it comes to urban trees are urban trees.
Yep.
As far as what goes along the side of the streets, a sycamore is actually a better street tree than a big leaf maple. The city made a mistake by planting hundreds of big leaf maples at the same time. And when the Columbus, and they don't do well with their roots constrained. And as the streets come up, they get old and big at 40 and 50 years. And then and then along comes the windstorm, and this is what happened in '62, and the whole town was in serious danger because the streets were completely blocked by all of the maples that had fallen down. I mean, ambulances couldn't get through. There was nothing. There needed to be some diversity of the street trees. And even though big leaf maple is native, it's actually not a good street tree. And that was that was the argument about the giant sequoia, is that it is a big a vigorous, hardy street tree. But even there, you have to have in a plan, like they don't last forever.
Mm-hmm.
And that kind of long-term planning goes beyond one generation.
I don't know. I have a lot of troubles with the sequoia, including the fact that they make your yard useless for growing anything else.
Oh yeah. Well, they shouldn't. They're not. Yeah, they're not yard trees.
No.
I mean, Douglas fir isn't either.
Nope.
Nor is big leaf maple. I mean, I started out in botany as a gardener in Seattle, and I remember more than once somebody would say, "Can you help my lawn?" This is the Pacific Northwest, which really isn't good for lawns. And I said, "Yep, I can probably get your lawn in pretty good shape. Cut that maple down," because they come in as volunteers, and within 20 years, their lawn is just a soggy mess.
On the other hand, we just took out our lawn and planted all natives.
Yeah. Yeah. Well, we're headed sort of the other way. We've got a vegetable garden where there used to be ornamentals.
More questions, Emmeline?
It looks like the questions are finally wrapping up. So great. And there's also, if anyone gets a chance, you can check out the chat. This video here is going to be available on our YouTube channel, the Native Plant Society of Oregon Emerald Chapter YouTube channel. So anyone can come back and watch it or hop in the chat if they want to. And thank you to all three of you for this very interesting talk today. I feel like there's quite a lot of ground that we covered, and it's so good to hear about such a local gem, someplace that I'm sure is really near and dear to all of our hearts. So thank you so much.
Pleasure.
Thank you.
Thanks for organizing. Thanks a lot.
It was a good motivator.
Yeah, it was certainly a motivator for me as well. So thank you so much to all three of you. Just a reminder, check out those and like stay tuned for our December social and for our January program on the Darlingtonia. And keep coming to these online Zoom programs. Check out our live things too. And if anyone wants to get involved in native plant conservation, that's totally an option. Contact us. So thank you all so much. Have a wonderful night.
Okay, great, thanks. This was fun. I just, in the last 3 seconds, finally figured out how to get my screen back.